This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 225, recorded on March 20th, 2013. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today right here in the TWiV studio in New York City, Rich Condit.
Hello, Vincent.
Rich,
how are you?
Probably everyone thought I was going to say Dick Despommier.
Yeah, where the hell is Dixon?
Oh, he's off traveling somewhere, as usual. How are you, Rich?
I'm outstanding.
We have some nice weather for you here, see?
It's beautiful. Is this actually the first day of spring, or is that tomorrow?
I think it's today. No, today's the 20th. It must be tomorrow.
Because people are talking about spring.
Well, it's almost here, but it just snowed on Monday.
Uh-huh.
Right now, it's very nice. Lovely weather. It's got blue skies with some clouds and 7 degrees Celsius. Also joining us today from Ann Arbor, Michigan, Kathy Spindler.
Hi, Rich. Hi, Vincent.
How are you today?
Doing okay, but our weather is not so pleasant. -1 Celsius.
Wow, it's chilly.
Yeah, 29 Fahrenheit. Oh, it just went to -2. Wow. But I think that's the last of the cold, because the rest of the week it goes up to 4 degrees, 5 degrees Celsius, and we'll be warmer.
I was up in Rochester on Monday, and it started snowing in the morning. Snowed all day, and it was tough flying out of there. It was quite delayed.
Yeah.
Now, we are recording on a Wednesday, which is very unusual for TWiV, because Rich is here in New York City teaching a course, a lecture in a course at Rockefeller University. So he did this once before, I think. You came up, right?
I have done this. Yeah, I did this, coupled that lecture with a TWiV, yes, once before.
I think Dixon was here last time.
He was. It was the first time I had met Dixon.
But there's no one here for you to meet this time. I'm sorry.
It's okay.
And Alan couldn't make it, so it's the three of us.
So I have checked a website called The Old Farmer's Almanac, and it says the first day of spring, the vernal equinox, 7:02 a.m. Eastern Daylight Time on March 20th.
All right.
So it's already happened.
That's why people are talking about spring.
Why do I think it's the 21st all the time?
Because sometimes it is.
Sometimes, not all the time.
Right.
Well, Kathy, you need to check these things, because apparently I get in a lot of trouble for saying all of us at some point or another. We get emails about this stuff. So.
Well, that's okay. People correct us. That's
I don't mind.
Part of what's going on.
In fact, one thing I would like to do before we proceed with this all-email episode, by the way, folks. I hear the switch is turning off now. They're already shutting down.
Oh, no. All email is good.
It's fun.
I wanted to follow up on one of the papers we did last week, the identification of the coronavirus EMC receptor. I kind of, I think we should read the authors from now on. We kind of went over them. I also didn't properly acknowledge the where the work was done. So the authors of this paper, published in Nature, the paper is called Dipeptidyl Peptidase 4 is a functional receptor for the emerging human coronavirus EMC. Raj, Mu, Smits, Deckers, Mueller, Dykman, Muth, Demers, Zaki, Fouchier, Thiel, Drosten, Rotier, Osterhaus, Bosch, and Hagmans. And the affiliations are the Erasmus Medical Center in Rotterdam. You have Utrecht University, Viroclinics Biosciences, Erasmus Medical Center again. Then we have the University of Bonn Medical Center, St. Gallen, the Institute of Immunobiology, also Saudi Arabia Virology Laboratory, and the University of Zurich. And I understand that the main drivers on this were the Drosten Lab, which is in Bonn. I kind of said last time, Fouchier Lab, not quite right. The Drosten Lab was also the first to put the sequence of this virus online. And they were also the drivers on the bat paramyxovirus paper that we did. I don't know if you guys
That's
remember
right.
that.
Yeah, I remember that. Yeah.
So
They sort of deep sequencing a whole bunch of paramyxoviruses out of bats, right?
So Christian Drosten is the head of that lab in Bonn. So it's a collaboration with, of course, other laboratories. The other paper we talked about a couple of weeks ago in MBio, which showed that ACE2 was not the receptor for EMC coronavirus, also from the Drosten lab, again in collaboration with Fouchier and others. So just want to get that straight.
Good.
Okay.
Good.
We have some follow-up. The first one is from Stephen. He sent a little note about a planet of viruses, which I had suggested to one of our listeners last time as a elementary virology book. He actually sends a little review by Michael Emmerman, or a piece of it. He says it's a very easy 2-hour read for anyone with a minimum amount of biology, say high school level. But he does point out some factual errors in the book, which he didn't particularly like. That is Michael Emmerman, who's a virologist at the Fred Hutchinson Cancer Research Center in Seattle. And I guess there's a link to it here as well. So you guys could look at that, you listeners, I mean, you could look at that review, and particularly the listener who wanted to have a book, and see if you still want to read it. I think it's still probably worthwhile.
Sure.
But perhaps consider the other one. Remind us, Kathy, what was the other book?
It's How Pathogenic Viruses Work,
Right.
And it's by Lauren Sompirak.
Right, that's a good one,
Sompirak.
too. And since he's a virologist, presumably there will be fewer errors.
Right. And he's updated it. I'm not sure when Zimmer's was written, but yeah.
Zimmer's was actually written in collaboration with, well, he wrote it, but he got a grant through the University of Nebraska Virology Center. When I was out there visiting last year, they told me that they have some grant for public education of science, and part of it was used to pay Zimmer to write that book. So there you go. Kathy, can you take the next one from Stephen? Same Stephen.
The following up yet again, that one. Yeah. Following up yet again on the term protozoa. Protozoa was a term I heard back in the 60s to refer to single-celled organisms that were animal-like, which is to say that they swam freely, didn't have chloroplasts, and ate their food, except that some of them do have chloroplasts. The term protist makes no such distinction, but is just a term for mostly single-celled organisms. And then he gives a link to a Wikipedia article about protists. Currently, the term protist, and I guess this is quoting from Wikipedia, currently the term protist is used to refer to unicellular eukaryotes that either exist as independent cells or, if they occur in colonies, do not show differentiation into tissues. The term protozoa is used to refer to heterotrophic species of protists that do not form filaments. These terms are not used in current taxonomy and are retained only as convenient ways to refer to these organisms. I don't know exactly how modern biology textbooks deal with this. So
This gets mighty confusing.
yes.
Yeah, clearly.
I can't keep this in my head. Nomenclature is always a problem.
Yeah.
I do like protist.
Yeah.
I've always liked that. But obviously, we had a good answer from Ricky last week too, right?
Mm-hmm.
About this. All right, maybe I should take the next one, since it was my saying that provoked this. This is from Neil, who writes, "Dear Vincent, I'm a loyal TWiV listener and huge fan, but today I'm writing with a small correction. About 38 minutes into TWiV 220, you mention that a Viron tried but failed to develop an engineered influenza vaccine and ended up licensing and marketing Masab's cold-adapted strains instead because, quote, that they couldn't come up with something on their own, unquote." Yes, I did say that. I couldn't help but respond, since that was my project for a couple of years.
Ouch.
To engineer, I'll refrain from using the RG terminology, thank you, an infectious, safe, and immunogenic master donor strain for influenza A. The details are actually interesting. It gets into genetic stability of attenuated viruses, how viruses are much more adaptable than we think, and ways to get around that. And he gives us 2 references for that. But the reality is that the CA strain that's Masab's cold-adapted strain, was made available for licensing right around the time that my engineered strain were being considered for advancement into the clinic. For several reasons, some of them business-related, the company decided to license the CA strains, and this then instantly became their lead product, eventually becoming what is marketed as Flu Mist today. The engineered flu vaccine program then evolved into an attempt to fill the niche where the CA vaccine doesn't work as well, which meant primarily targeting the elderly. Flu Mist doesn't work great in the elderly because they have more cross-reactive immunity from multiple previous infections, vaccinations. However, the challenge of balancing safety and immunogenicity in this population was significant, and eventually the company decided to terminate the project. I worked on parainfluenza virus 3 for a while, but eventually left to join a company working on HIV drug resistance diagnostic assays where the theme of adaptability is also highly relevant. At some point, I'll write about another pet peeve, predictions that some new antiviral will be resistant because it targets the host, not the virus, but not now. Anyway, I'm trying not to be overly defensive, but I do think that the choice of words was unfortunate. Aviron did come up with something on their own, but the CA strains became available and had already been in clinical trials, and so leapfrogged the internally developed candidates. Your mentor, Peter Palese, who was on the SAB of Aviron while I was there, can certainly give you more details about this than I can. In fact, the long and winding story of the development of Flu Mist might make for a good topic for TWiV one day. Thanks to you and the crew for the great service you provide. I consider myself a TWiV evangelist and tell anyone who is interested to listen as well as whenever I get the chance. Best regards, Neil.
So, yes, I agree that the long and winding road to the development of Flu Mist would be a great topic. We have talked many times about getting Harry Greenberg on here, and he can talk both about that because he was a principal in Aviron and about gastroenteritis, which would be great.
Okay. I guess Harry would be the guy to do it, right?
Harry's the guy. Right. Well, he is a guy.
I think we should get Neil on because Neil has a good, interesting career, right?
Yes.
And he can also talk about antiviral resistance. So, Neil, let's hook up and we'll figure out a time. All right. Rich.
Ricardo writes, "Hello, TWiV gang, the ones who make their listeners wiser so they keep coming for more. Just another example of parasites controlling behavior of their hosts." And he provides a link to a... I had this now. I'm going to have to do it again. Hang on. I'm left-handed here, so I'm having a bit of a problem. Open link.
How is your collarbone, by the way?
It doesn't hurt. It doesn't hurt. It's just inconvenient. That's all.
We did mention it on TWiV.
You did mention it on TWiV a couple of weeks ago, just after it happened. It is on the mend. I'm doing okay. I'm just a little late. It's to this is to an article from National Geographic, and the article is titled "Parasites Make Their Hosts Sociable So They Don't Get Eaten." Ah, yes, I remember this. This is a parasite of a brine shrimp. And what was the parasite? A microsporidian,
right.
a fungus,
yeah.
Microsporidian that makes these parasites swarm. I mean, makes the shrimp swarm, and that makes it so that that's good for the parasites because it keeps them in closer proximity, and they can be handed back and forth. And there are other advantages as well.
Yeah.
So this is. One of several examples of microbe infections altering behavior of hosts.
Now, you would think this would be a TWiP topic, but he sent it to TWiV. He said, "Hello, TWiV gang," so I put it here. I guess TWiV listeners might be interested as well, right? Why not? I think it's very cool. He also sent another one, which this is right up your alley, Rich. I don't know if you can see it there. First page on one of our local newspapers. It's a Nature. It's a link to a Nature article.
Oh, yes.
It's JX594. Does that ring a bell?
Yes, and I actually looked this up. And did this come up in this email or one later where somebody was asking about the mechanistic logic of some of these. So,
it did come up later.
yes.
Yeah.
So this is yet another clinical trial of an oncolytic vaccinia virus. And I didn't actually look at this one closely, but suffice it to say that these are showing significant progress. And when we get to the subsequent letter, perhaps we can talk about mechanism and stuff. I did look a little of that up.
Yeah, this was a trial in people. It's a dose-finding trial. But they said it was oncolytic, and the tumors responded.
I'm getting half a dozen papers a week.
Yeah.
Of poxvirus oncolytic stuff of one sort or another. So these are being pursued vigorously as potential cancer therapies.
Right. There is another one here from Neil. It's the same Neil, I believe, that we just heard from, who writes, just wanted to suggest an alternative to SurveyMonkey that you were discussing in a recent episode. I've used it now and then, but have mostly moved over to Google Docs, where you can create a form and have the responses go straight to a spreadsheet. As far as I know, there is no limit to the number of responses you can collect. I use it to help manage the recreational hockey league I run, that John Boothroyd, previous guest on TWiV, also plays in. How about that? You can split the form up into multiple pages, have users directed to certain pages based on their responses, and so on. Plus, you can share the online results spreadsheet with whoever you choose, transfer to Excel, etc. That sounds cool. I didn't know that. Love the podcast. One of these days I'll get caught up so I can email about my questions about the most recent one and have them read on the one after. But for now, I'm too far behind. If you ever feel like interviewing someone who has had academic training, biotech industry experience, a short stint at public health at WHO, and now is an independent consultant, talk about alternative careers in science, I'd be thrilled to do it. I've worked on influenza, HIV, and HCV in the vaccines, antivirals, and diagnostic assay areas. Neil. So that's the Neil who wrote the FluMist letter.
Right.
That's why I was saying he'd be a good guest.
Mm-hmm.
Okay, that brings us to Kathy.
Actually, just
yes,
a second.
sir.
I want to go back to that.
Oncolytic?
No, the article before it about the microsporidium that infects the shrimp, because I kind of fumbled that a little bit. That and apparently a tapeworm infection as well will make these shrimp swarm. And the idea is that because they swarm, they are more likely to get eaten by various predators, and that helps them spread because they get spread in feces. Okay,
I see.
So pretty weird.
Interesting.
All right,
Kathy, you're next with
David.
Yes, please.
Dear Vincent, Racaniello, Dixon, Despommier, and everyone else at TWiV, I am a biochemistry student at Skidmore College in Saratoga Springs, New York, and for my virology class, our professor has been assigning us to listen to your podcast every week. And so once a week, I have been going for long walks in the nearby woods while listening to your podcasts. But I wanted to know if you could discuss a topic I came across while surfing the internet, the use of engineered viruses to fight cancer cells, and he provides a link. Would it be possible to discuss the biological mechanisms behind the engineered virus fighting the cancer cells while boosting the body's own immune response to the cancer cells? What are the key differences between the engineered viruses versus the natural viruses? Given how specific the viruses attacked the tumor cells, this definitely opens a whole new world to cancer treatment and possibly even the treatment of other diseases. Thank you so much, David.
It's the same paper, right?
Yes, it's the same paper.
Okay.
And this is the paper is, did we give the title, Randomized Dose-Finding Clinical Trial of Oncolytic Immunotherapeutic Vaccinia JX594 in Liver Cancer.
So we've had a couple of oncolytic episodes, right? We had Grant on once to talk about Grant McFadden.
Right.
And then we had the CEO of, what was it, Oncolytics? Is that the name of the company in Canada?
So. It's a real virus company.
So maybe, David, you would take a look at those. But I'm sure we'll do it again.
There are I looked up this Generex 594. I had looked at this before, but there's I found another link where the abstract gives a good mechanistic description of how the or at least theoretically, how the engineering of that particular vector makes it specific for cancer cells. Mm-hmm. And it actually has two significant changes in it. One is the thymidine kinase gene is knocked out, and the other is that they've knocked in expression of a granulocyte macrophage colony stimulating factor, which is an immunostimulatory molecule. And the idea behind the TK, it's been known for a lot of years that, so thymidine kinase takes a DNA precursor, thymidine, and phosphorylates it, and without that phosphorylation, it can't be used. So it's a salvage pathway. It's not absolutely critical for the virus replication, but it assists, and it's been known for some time that if you knock that out of the virus can still grow in culture, but it's attenuated. It's less virulent in vivo. So if you want to make a safer vector or something like that from vaccinia, knocking out the thymidine kinase makes sense. Now, cancer cells replicate in an uncontrolled fashion, and part of that is boosting nucleotide metabolism. So they are likely to make more of their own thymidine kinase, and so the virus might, a TK minus virus might replicate better in cancer cells, okay, because they supply a thymidine kinase. So it might have that might give it some targeted specificity, replication better in the cancer cells. And the expression of the GM-CSF tweaks the immune system so that it's more directed towards the cells that are expressing that molecule. So those are just two examples of ways that a virus vector could be engineered to target cancer cells.
Another way is the way that people in the adenovirus field have looked at this, and I had picked a paper by Claudia O'Shea's before that wasn't directly related to this oncolytics question, but she's been active in that field. And the principle there is that you have viruses that are unable to inactivate cell checkpoints, and that then those viruses could selectively replicate in tumor cells in which those checkpoints are already inactivated. So those viruses would have a disadvantage in a normal cell but could have an advantage in the tumor cell. That hasn't always played out exactly like people thought it would. And so, in fact, for the adenovirus vector that's most famous, Onyx 015 or DL1520 that Doug Barker made in Arnie Burke's lab, it turns out that it's not the inactivation of P53 by this mutant, but it's another function of the E1B55K protein having to do with RNA transport out of the nucleus. So there are a lot of mechanisms, and maybe we need to hear some more of those at some point.
So this takes me, in my mind, back to a TWiV that we had with Patrick Moore on cancer-causing viruses. And he made a point that I had not considered before, which at least my own sort of spin on this is that there may be significant overlap between cellular pathways that are suicide pathways that are checkpoints for errors in DNA replication and that kind of stuff. So things that lead to apoptosis when replication goes wrong. There may be significant overlap between those mechanisms and a lot of antiviral mechanisms. And so viruses may not do well on normal cells because their antiviral mechanisms are intact, and they may do better, grow better on cancer cells because those cancer cells are defective in these checkpoint pathways, which are the very same or at least overlapping with the antiviral pathways. That may become a general phenomenon.
Yep. So TWiV 131 was a rheostat for cancer, and that actually we have a transcript of that episode. So David, you might want to check that out. There's a lot of I'm looking at the show notes. They're pretty cool. There's a lot of good stuff there. We have some articles on Onyx 411. Then there's Oncovex, GM-CSF, and the reovirus. The reovirus is actually Terry Dermody was here last week, and it's just type 3 Deering strain of reovirus, which in this case, targets tumors because of their weakened innate responses.
Right. Same thing.
So it's pretty cool. But yeah, this David, you might not have found some of these, so check them out. I don't know which one is the Grant McFadden one, but we'll have O'Shea on at some point. She can talk about it as well.
The other thing I liked about this is that David is out there getting exercise while he's getting TWiV.
Cool.
So that's good. Yeah.
We're good for body and mind.
You bet. Rich, can you take Monica, please?
Sure. Monica writes, "Hi, just a quick message to let you know I love your podcast. Keep it up. There is valuable information to share. Thanks, Monica." Short and sweet. Very nice. Thank you, Monica.
Why don't you take the next one, too,
Okay.
since it's so short.
Ann writes, "Hi, TWiV crew. I am a graduate student in virology with an eager interest in science communication and advocacy. I would love to draw everyone's attention to a new campaign, quote, Ask for Evidence, unquote, that was launched in Boston last week, springboarding off of the AAAS conference. As a brief background, the UK-based non-profit group Sense About Science equips people to make sense of science and evidence on issues that matter to society. They run several campaigns to raise public interest and even activism in several fields. See their website for details, and she gives a link to their website. A few ambitious young scientists have brought Ask for Evidence campaign to the U.S. The goal is to encourage the public to be skeptical of the claims they hear in advertisement, the media, and in politics. The website provides support for inquisitive minds, how to phrase a request for evidence, how to evaluate an evidence that is provided, for example, is it published, peer-reviewed, and a supportive community to help follow through with holding the company or media outlet or politician accountable. And she provides another link for that. Think of the folks curious about home remedies, detox treatments, and dietary supplements. Think of patients with chronic diseases with no treatment options remaining, trying to sort through the myriad of miracle cures advertised online. Not only money, but time and health are on the line. It is critical for the public to understand the scientific evidence, or lack thereof, that supports consumer products, policy, and so forth, and an alert and proactive public will promote a culture of accountability. I'm excited to see scientists and lay skeptics stepping up to lead this movement. Thanks for the consideration. As always, I send my sincere appreciation for the wonderful work you share with us all. Best wishes, Anne." So, terrific. This looks like a terrific movement. You know, one of the things I think about is, Snopes? Anytime there ought to be a scientific equivalent. Anytime I hear something that sounds like an urban rumor or an urban legend, I look it up on Snopes and find out whether or not it's true. There ought to be a science equivalent, but this provides more like tools about how to think about stuff, which would be great.
So, Kathy, what's this about a button?
Well, so two different things. So on the site, they have a couple different sections, and one of them was a Don't Destroy Research section that included a video where 4 of the researchers involved in a long-standing GMO trial made a YouTube pleading, essentially, with the protesters, don't destroy our research. If you want these answers, this is the best way to get them. And evidently that was effective. So that was worth looking at. And then in this newer Ask for the Evidence section that Anne referred to, they have a campaign to have people include a button on their website, and they list and show various people doing this with this button. And so if you've promoted this on your website or your podcast, you can potentially get a link on their page. So I thought that would be a way to get TWiV there.
They should have TWiV here.
Yeah.
I mean,
so
really.
I think you have to get the button, print that out on cardboard, hold it up, get your picture taken. I don't know how you get that cardboard thing, but anyway.
But there's I'm clicking all over here, and I'm not getting anywhere to these people's websites. So, I mean, I'd be happy to put a button for their website on ours, but I think they should have one if they want to inform people, which is part of their mission, right?
Right. Well, so at the very bottom of that link that I provided, the A4E one, then they list a couple of the podcasts. So I think you'll get there.
All right, I'll look into that. That just gave me an idea. 2, to put something on. Yeah, here we go. It's their links at the bottom, yeah. Selected coverage. I was thinking we could put a little code on TWiV, which people can embed in their website, and they'll have a TWiV button.
Right.
How's that? That's good, right?
That would be really good.
Because I just haven't done that, but this just gave me the idea. So There you go. But one of the things, this is great, but I think they should point people to other sources of good science information. And maybe they're doing that a little bit here, but they could do more. So I'll look into that.
Well, good for them. I hope the difficult thing, it seems to me, is going to be to make this both prominent enough and easy enough to use so that the same people who are going to surf the web for anecdotes about this or that issue are also going to find this and use it appropriately. Okay, I think visibility and manageability are issues, and I hope they're successful with this because it's a great idea.
I have a letter from Paul Nurse, who used to be president of Rockefeller, and but they don't have a picture of him. He didn't send them a picture with a cardboard ask for evidence button. Guess he didn't have time. Okay, that brings us to Ken, who writes, I stumbled upon this article early today, and he sends a link to an article in The Guardian, and a number of listeners have sent us links to this story. U.S. doctors cure child born with HIV. And he says, I remembered you guys talked about the eradication of viral diseases, specifically HIV, through prevention when Dr. Anthony Fauci was on the show. The article talks about the first case of cured HIV through the use of an aggressive treatment in an infant by bombarding the infant's system with antivirals. Will this kind of treatment be viable for the eradication of the virus if all babies born with HIV were to be treated with this method? Could there be any kind of complications if this method was used to eradicate the virus? What do you think of this method? I'm a high schooler in California and sadly not much of an avid listener of TWiV, simply because I do not have enough time to listen to most of the broadcasts. However, I do in fact love TWiV as it gives me more knowledge on virology and medicine than the high school curriculum does, and will definitely catch up on episodes after I am granted my freedom. Man, you go to college, you're going to be less free.
Depending on what's imprisoning him right now. Who knows?
I thought it was high school.
Could be.
Anyway, this is a very interesting story, which I've been looking into and asking everyone about. But the problem is there is no paper. There's no journal article.
Right. We need to see the primary data.
And we need to do that before.
Somewhere along here, there is a link to the conference.
Oh, yeah, the CROI.
I did not have a chance to look at that whole thing. Did either of you guys?
No, I mean, I didn't. It's pretty much what the press has been summarizing that press conference or that report, and I would like to see data. I mean, the story is that a baby was born to an HIV-positive mother, and the baby was apparently HIV positive, but, we don't have the data, so we can't evaluate it. And then they aggressively treated the baby with antiretrovirals 30 hours after birth.
And apparently the normal treatment is not the full course of heart treatment. It's like 2 antivirals or something rather than 3. Is that right?
Don't know.
And they were more than, at any rate, they were more than usually aggressive in this case.
They were very aggressive, and it was not a continuous therapy. It was a defined therapy for a defined amount of time, and now the child is 2 years old or something and is free of infection. But, in adults, this is very difficult to do because you have all these long-lived reservoirs of infected cells, latently infected. You can't get rid of them, and their half-life is infinite, as I was told in Rochester by an HIV person. I asked, what's the half-life? He said infinite. So some of these cells live forever. So by treating them with antiretrovirals, you can't get rid of the DNA inside the cell. So, and maybe in a baby, there's just not a big reservoir. Did you talk to your colleague there who works on the long-lived reservoir?
Oh, I did not.
I
did not. Kathy Collins.
Yeah, who we had on TWiV. It would be interesting to know her take.
Got her spin on this.
But I think we have to wait for the paper to be published.
Absolutely.
Right?
Exactly. Yeah.
Yeah.
And even in this article, though, it says that the physician who cared for the child said, this is the first functional cure of an HIV-infected child. But then the writer goes on to say, a patient is functionally cured of HIV when standard tests are negative for the virus, but it is likely that a tiny amount remains in their body. And, we just don't know if this patient is now 2 or 3 years old. What's going to happen when they're 45 or 60 or something, There's just a lot of unknowns here.
I mean, the treatment itself may have problems when this child is older. Who knows, right?
Right.
It's not clear that it's good to do that. Of course, the alternative is to have HIV, but we really have to see. I don't think this is something you could apply to adults. We know this doesn't work, but maybe in children. Apparently, there aren't so many of these children born in the U.S., but there are many, many outside of the U.S., so this could be useful for babies, I suppose.
Now, somehow looking over this stuff, I came up with a paper on treating adults and getting close to getting some long-term effects. Is that a letter coming later on, or did I find this on myself?
I think there's another. I think there is another email,
so we have
about
more
this.
discussion about this later on.
Yeah. Are you next, Kathy?
I think so.
Yes, I think.
Trudy writes, Dear TWiVers, I'm a little behind on my episodes and was listening to TWiV 218 on my way to work this morning. I apologize if this has already been addressed, but after hearing about the little episode with Welkin and his daughter's flu inoculation in the eye, which of course was accidental, as well as the discussion about the possibility of infection through the mucosa of the eye, I looked up some of the publications of a friend of mine at CDC. Please see below. Of course, these studies are in ferrets, and by now we all know very well that ferrets are not human.
Excellent.
Yes.
So these are about infecting ferrets in the eye with influenza, right? Mm-hmm. Both papers.
Right.
You can do it.
Right.
You can infect them in the eye and wind up with respiratory symptoms.
So what's the path for the virus?
I was thinking about that. I don't know. Don't the tear ducts drain into the nasal cavity and then couldn't you get it that way? I don't know. I meant to ask an MD. But I mean, think about, when you cry and then, then your nose runs. And I always think that's from something from the tears. But I could be totally wacko. I'm not that kind of doctor.
Yeah, I think we have some docs listening. So tell us.
Yeah, we'll get some follow-up on it.
Tell us how putting influenza in the eye would lead to virus in the respiratory tract, assuming it's not just coming down in tears and draining down your face into your nose. Okay, that's not what.
Right. Yeah. No, I know. I don't mean on the outside. I meant
inside.
On the inside. Yeah.
And this has got to, of course, be maybe what we need is a veterinarian, right? Because we're talking ferrets here.
Right.
Of course, because they're not human.
Right.
That's right. Well, maybe some of the anatomy is similar. I don't know. I know some human ferrets. Do you?
Weasels.
No, weasels, not ferrets. That's right.
Different thing.
Are they related?
I think so. I would. Oh, now you're
going to get in trouble.
That's right.
Let's move on to Shane. I think it's my turn, isn't it? No, it's Rich's turn.
I think it's my turn, and that's good because this says, Shane says, for some odd reason, hi, Rich et al.
That's okay.
Oops.
He wants to just mix it up,
Yeah. I'm catching up on all the TWiV, TWIP, TWIM podcasts, and I'm still quite far behind, but a couple of months ago, started from TWiV number 1, and am following in date order. I love them all, and I'm not looking forward to the day when I need to slow down and listen at a more leisurely pace. In TWiV 133, you mentioned that biological control methods are doomed, and Dick agreed with you. The Cactoblathus moss was introduced in Australia to control the spread of prickly pear, and although it hasn't eradicated the plant, it has been successful in controlling it to a level where native plants have been able to reclaim land that was previously claimed by the prickly pear. Keep up the great work, you guys. Kind regards, Shane. So we probably said that biological control methods were doomed because, the target of the biological control is going to bounce back with resistance of one sort or another. If you look at the Myxoma story, right?
Yeah.
Myxoma wiped out the rabbits, and then the rabbits became resistant and that kind of stuff. So let's keep an eye on the Cactoblastus moth and the prickly pear and see if that pans out.
That's a cool story.
Yeah, it is a good story.
Well, you cannot be absolute in science. That's the thing.
Right.
So we shouldn't say it's doomed. We should just say it doesn't look good. But, it turns out that TWiV 133, Kathy, is our episode with Kathy Collins.
Ah.
How about that? Is that coincidence?
That is.
Huh. I guess we were talking about control in that episode. The next one is from Robin. Is it my turn now?
Yes, it's your turn.
Robin writes, several thoughts. Speaking of infectious disease and ecology, the matter of attenuation could be looked at from the perspective of fitness for the pathogen. Allowing hosts to survive helps, as does enhancing transmissibility. Promiscuity in selection of hosts also works towards this end. Transmissibility is also enhanced by greater replicability, often at the cost of host survivability, as in smallpox. Incorporation into the germline is the acme of transmissibility, for which the sine qua non is survivability of the host, while also limiting replicability, as in the case of the endogenous retroviruses. Promiscuity in host selection, as pointed out by the sage Dixon on his This Week, is exemplified by toxoplasma. Perhaps the phenomena of attenuation versus enhanced virulence could be studied from this perspective. I think we were talking about attenuation and fitness and transmissibility at some point. Sounds like it. Mm-hmm. Well, these are all reasonable thoughts, but, making rules is hard because there's always an exception. I think there's an email later on where someone talks about transmissibility and virulence. But I don't know if Robin's talking about attenuation from the perspective of natural attenuation, or I thought it was about vaccines initially, but I don't think it is.
No,
right?
This, to me, what I get out of this is that there's some sort of, depending on the circumstances, balance between how pathogenic an agent is, no matter how much damage it does to its host, and its transmissibility. And the worst thing you can do, a bad thing you could do as a pathogen is to kill your host too fast. You'd kind of like it to hang on for a while and spread you around, okay? But then there may be, under those circumstances, you may not make as much pathogen in the per animal. I don't know. So there's some sort of balance between virulence and transmissibility that probably varies depending on what pathogen-host interaction you're talking about.
So smallpox is one of the more virulent, highly transmissible pathogens, right?
Yeah, and one of the things that we teach is, or that I have taught, is this idea that zoonoses represent non-equilibrium infections. Okay,
right.
So a virus coming from some other animal gets into humans, and it's not adapted right. And quite often, what happens is you get a lot of virulence and not much transmissibility. And if it manages to get a foothold in a new host, what you find is that over time it reaches an equilibrium till you may reach a point where you have an agent that really doesn't cause much damage to the host. You may not even know you're infected, like most of the polyoma viruses infect us, and we never even know it. Interestingly, smallpox was really obnoxious. At the time when vaccination was being introduced, Variola minor, or elastrum, was emerging in South America, I think, and Latin America, and that was a much less virulent form of the smallpox virus, and only had about a 1% mortality or something instead of a 30% mortality, and yet was cross-reactive. So if you were immune to alastrim, you were immune to smallpox. And I've often wondered whether this didn't represent a step towards equilibration of the of variola with humans, and in the absence of vaccination, if we may have reached a point ultimately where smallpox wasn't as big a problem because alastrim or something even milder took over.
All right, Robin continues. The root word unguis means nail. I guess we're talking about ungulates.
Ungulates, right? Yeah. This is a enormously entertaining email because she's obviously made a list of questions or screw-ups of ours, and she's going through one at a time and straightening us out. I like this.
It's a guy.
Oh, it's a guy. I'm sorry.
That's all right. The ungulates are a group of mammals in which the unguis has been modified into hooves. Artiodactyla, which comes from the Greek, and I'm not reading it, forget it. So the name even-toed is a translation of the description. So Artiodactyla are the ungulates with cloven hoofs. They are not actually cloven in the implication that it was originally one hoof, because the digits involved have always been phylogenetically separate. They correspond to our middle and ring fingers. In humans, the commonest form of syndactyly involves the same digits in the hands and feet. Some of these animals have become ruminants, chewing the cud, thus processing it for their internal fermentation tank. They are the ruminants. In Mosaic law, the ruminants and birds are the only kosher terrestrial vertebrates. The largest of these is the giraffe. Tradition has it that the appearance of a ruminating pig will make it kosher and herald the appearance of the Messiah. Messiach. Diptera, two-winged, is the insect order that includes flies, mosquitoes, and midges. Brachynathia inferior is present in humans in the Pierre-Robin syndrome. Brachynathia. When did we talk about that?
We talked about ungulates and ruminants with respect to the Schmallenberg virus. I think when Dixon was on.
That's right. But brachynathia, I don't understand that. This is a malformation of the...
Malformation that gives you a small jaw and other issues.
Huh.
Oh, there was... I'm thinking there was the goat, it was a sheep, also in the Schmallenberg. Was that a Schmallenberg paper? Where they had the picture?
Yeah. Maybe that's it. It's malformed, yeah.
Yeah.
And we mentioned that word, and we're probably puzzling over it.
So, giraffes are kosher.
Apparently.
That's good to know.
But who eats giraffes?
Nobody that I know of.
No.
Maybe in some countries they do, but they're not easy to capture, right?
No, they're really skittish, kind of.
All right, thank you for that, Robin. Whose turn is it next? I forgot.
Was that you, Vincent? It's Kathy, because we're going clockwise. Kathy's the
All right, Kathy.
Okay. Liam writes, "Hi, Vince et al. Disclaimer. This email got a little out of hand at over 600 words. At this length, I thought you might be more comfortable reading it structured like a paper, so here you go. Abstract. I'm a neuroscientist who has been listening to your show on and off for 2 or 3 years, and I've decided to start my own podcast in a similar vein. Here, I ask questions about the development and technical aspects of your podcast and present 2 possible picks of the week. Introduction. I started listening to TWiV a couple years ago when I was doing my honors project in a virology lab at the University of Guelph. I've always been more of a geneticist at heart, and I'm now working on my PhD in a neuroscience psychiatric genetics lab at McGill University in Montreal. But I've been an occasional listener of the show ever since. I've always been really impressed by the way you balance your show, making sure it is informative and interesting to experts, but still appeals to non-experts. Since virology is a small field, at least in comparison to genetics or neuroscience, I assumed other fields would have similar podcasts. I've looked high and low and have been pretty consistently disappointed by what I have found. The best case is interviews with authors unbiasedly brought to you by the journal that published the paper. So I've talked to a couple of my talking adept friends and colleagues, and we're going to impart any advice and we're going to start our own. I have a little bit of experience in radio, but short story long, I was wondering if you could impart any advice on things to do or avoid that you've gained over the years of podcasting. Results. There are a couple of specific areas in which I particularly appreciate your advice, but feel free to add on anything else you think would be helpful. First and foremost, I'm really impressed by the size and diversity of audience that you've accrued, and I was wondering if you have tips for keeping things interesting to the grad student researcher audience, but still accessible to outside groups. In the same vein, how much information do you have about your audience? I've been using you guys as an example of how successful this kind of format can be, but if I could include some actual numbers, it would make my job of pitching the idea a lot easier. Second, that's only the second point? Okay.
That's why you had to put it in paper format.
Right.
It got out of hand.
Do you approach papers you are using for the podcast differently from papers you are reading for your own research? I know that for me, so far, thinking about how to present a paper has changed how I write papers. I tend to look for the narrative element, but I'm curious how you approach it. Finally, I have a couple more technical questions. I went back and listened to some of your first shows recently, and I noticed that your audio quality has improved a lot since then. What do you use in terms of recording equipment? I'm also curious about your software, how you record from Skype, and what you do to make your final product. Finally, editing. I read some people talk about podcast editing who talk about spending more time editing than recording. I've done some music and interview editing, so I know that this is possible, but I'm not sure it's necessary. How much editing of the show do you do? Conclusions. I'd like to end with the pick of the week. Maybe we should stop there.
Yeah, okay.
Come back to the conclusions, because there's a lot of questions.
Okay. The first part was the size and diversity. How do we keep it interesting? So I think that we just pick cool stories that would appeal to a lot of people. Good science, so we pick from journals, so that means it would appeal to a student, right? Mm-hmm. A researcher. But dealing with things that are in the news, like the coronavirus receptor, mm-hmm. stuff like that, oncolytic therapy. I mean, now and then a little more obscure, like splicing in flu, but it's flu, so people mm-hmm. like to hear about flu. I mean, there are lots of papers out there. They're great, but they wouldn't cut it because it's details and it wouldn't really excite our general audience. So that, I think, is the key. And I always keep an eye out, and I think you guys too, for stuff that is really that would be interesting to non-scientists. What do you think?
Yeah, and I don't know. One of the reasons that it appeals, I think, is just your model for this all the time, that I refer to all the time, is that we have these scientific conversations in our office when people come through, where we're just talking. We're just talking about the science, and wouldn't it be nice if we could make these public? So there's nothing fancy.
No.
Okay? It's just a conversation. And I think we do keep in mind that we're talking to a general audience, so occasionally make the effort to have some explanation to sort of not dumb it down, but make it more accessible to a general audience. But beyond that, we're just doing what we do every day. Have a conversation about the stuff that's out there.
It's really conversational. That's the key. It's not lecturing.
Right. Mm-hmm. Right. And there's not a lot of, I mean, we get excited and stuff, but there's not a lot of gee whiz that goes on either. It's just a conversation, you know?
Mm-hmm. What do you think, Kathy? Is that about right?
I think that's about right. And, you know, any one show might appeal to more of one demographic than another, but the whole set of shows, you know, where we're doing Virology 101, or we're interviewing the scientists themselves about the work that they did, or a different kind where we're doing just talking about someone's paper but they're not on the show, or this where we're answering all kinds of email. You know, there's something in there for everybody, I think.
Yeah, that's a good point, to mix it up, not just do papers every week.
Right.
Yeah.
Oh, yeah. I think that's, this stuff with email and picks of the week and all that kind of stuff is important. Over time, Vincent, you've talked to a number of different people, and you've managed to identify people who like to talk, people who are conversationalists and can hold up a conversation, and that there's a chemistry here that works.
I was talking to the students at Rochester on Monday. They wanted to know. And I said, you know, I put together the team slowly, not immediately, because I wanted people to fit. So Dixon and I started, and we were having a great conversation. So I wanted people that would fit into that. And that's really important, because not everyone is comfortable just chatting in in a non-structured way.
Right.
Interestingly.
So what about this question of the numbers? The reason I'm asking is because today was the day for students to turn in the first of their extra credits, which are based on a paper that was discussed on a particular TWiV episode. So it's there's a potential that we have 35 new listeners, or at least have listened once.
Yeah.
But I wonder how often somebody listens once, and then if you have any idea with what frequency they convert to a more regular listener. Or
Yeah, I can tell you every day the number of downloads, what country they came from, which episode, what kind of computer they were using, what software. It goes down pretty far, but I don't know. I don't think I get their IP address, no. So I don't know if they're coming back. Well, I know that these are unique downloads, so we have a lot of detail about that, but I don't know if people are coming back. We did a survey some time ago. We got 1,000 people to respond, so we have from that a good sense of, what our listeners are doing in science, non-science, etc. So I think you have to do some kind of survey, but you're not going to get that right away because when you start, you're not going to have many people listening. But depending on where you put your podcast, you can easily get stats that comes with it. It's no problem.
He asks how we approach papers.
Oh yeah, that's good.
And personally, I don't think I've gotten better at reading papers because of TWiV. Okay, I'm quicker at it. All right, but I don't think my approach is a whole lot differently. The way I read a paper in general is to one way or another make sure that I understand if and how the data support the conclusions. All right? So I focus on the figures, and of course I read the introduction and the discussion, all that kind of stuff. But I usually get far enough into it so that at least on the key pieces of data, I feel comfortable with understanding how the experiments were done and whether or not the conclusions, how the conclusions from the data or how the data support the conclusions that they make in the paper. When I feel comfortable with that, I feel comfortable discussing it. And that's no different than I would do for a journal club or anything else.
So, Vincent, then the technical questions, do you have that somewhere on your blog or something where you talk about Skype or your software, your hardware, any of that?
I don't. I probably should summarize it in a document somewhere because I don't want to take the time to do it now. But I would like to say one thing, and that is the audio has to be really good because bad audio is a killer. All right, and I've learned that from listening to other podcasts. So I spent time learning how to make it good, and now I'm at the point where it sounds good. And there are tricks with Skype and so forth. But you have to put time in to make it good. Not a huge amount of time because then it will be you'll be inhibited from doing it if it takes you too much time. So great audio. The other thing is you have to be regular. You have to decide on a schedule and do it. Stick to it. Otherwise, if you don't have a schedule, that's the end of the podcast. Eventually, you say, "Oh, we're not going to do it this week," and then next week you don't do it. Then the following week, and then it's gone. So you got to pick a day of the week, a frequency, and stick to it. That is one of the most important components of having a successful podcast. So with Dixon, I can't make a schedule because he's not around that much. So we hardly release TWiPs. And other podcasts that I've listened to, they end after so many episodes. So
yeah, not only recording on a specific day so that the people who are working with you are regular and they know that they're going to do this on Fridays. But we have listeners who wait for these things to be posted, okay?
Yeah.
And if you disappoint them on a regular basis, you're going to lose your listeners.
Yeah, listeners do tend to be regular in when they download your content. And if it's not there, then they're going to stop listening. So, I release on Sunday, and I know people wait and they get it for Monday for their commute. So I don't want to disappoint them.
How many dollars do you, I'm looking at a lot of wires and knobs here in the TWiV studio. How much money do you have invested in this stuff, Vincent?
Well, we did have some advertising support a while ago from Go to My PC and from Drobo. So I think I brought in a few $1,000s, and I spent it all, and I've then put in a few thousand more of my own investment into this stuff. And I could do more. I know exactly what more I need to make this even better, but it's okay now, and I want to raise some money to do that. Now you can do it a lot less. I mean, when I first started, it wasn't that much, but I've added that over the years. You can start with a computer, and you need some kind of recording, and how you do that is it's not easy. But I did a pick of the week, the Podcaster's Studio, a while ago. It's our friend Ray from ASM who has his own podcast on how to do podcasts, and he's very good. If you want to listen to him too. But at some point, I'll put together a description of how I do it.
Editing.
Yeah, editing is as I said, you have to make the audio sound really good. So I do some post-recording editing, but I know exactly how to do it now. I mean, you could go through and take out every cough and throat clearing, but I can't do that anymore. It's too much time. It would double the listening time of the podcast. So if we recorded an hour and a half podcast, it would take me 3 hours to do that.
So what do you actually do?
Well, I have some tweaks that I use to sweeten up the sound. Cut the beginning and end off. I put the music in, and then I scroll through the waveforms and look for what look like coughs or something like that. I can tell those now by just the pattern of the waveform, and then I stop and I take those out. But I don't play the whole thing through because it just takes too much time. I just kind of go through it quickly.
So how long does it take you to edit like an hour and a half episode?
Probably half hour just to go through and edit it, and then I export it, and then making the website and all the other stuff takes other time. So it takes about an hour and a half to 2 hours to get the whole thing edited and posted.
Wow.
Yeah, it's a lot, and sometimes I have plenty of time, and other times there are other things. So
you mean you have other things to do?
Yeah.
Yeah.
Some other things.
On Sunday?
Yeah, there's usually something to do. But,
okay, so maybe that answers most of Liam's questions. I'll move on to the conclusions.
Mm-hmm. Sure.
I'd like to end with a pick of the week suggestion. I know you have discussed a couple of papers from the journal eLife, but the journal only started having its own site last month. I've been saying for years that one of the key steps for the open access movement would be to have open access journal in direct competition with journals like Nature. And eLife, despite its stupid name, seems like it might be that journal. The thing they've got me wondering about is how they will monetize the journal, since right now they're being supported by their backers. Another interesting service is called Rubric, which is RUBRIQ, which is designed to give fast peer review turnaround with reviews that will be accepted by a wide range of journals. I'm a little bit hesitant about the privatization of the peer review process, but what we have now is insanely inefficient, so I might be willing to give it a shot. Finally, I want to thank you guys for your contributions to the community. It's really impressive what you've accomplished, and if I can be a fraction as successful, I'll be ecstatic. Conflicts. The authors have no conflicts of interest to declare.
All right, that's nice.
Yeah.
Go for it. You'll do fine.
Yeah, just do it consistently and get some good people with you who will talk. Yeah.
Right.
Rich, can you take the next one?
Right. David writes, Dear TWiVologist, listening to your podcast has awakened in me a fascination with paleovirology. Indeed, starting with the information from several of your podcasts and Vincent's online lectures, I came up and gave an informal talk about paleovirology to the local fossil club. In doing web-based searches on paleovirology, I came across one paper different from the rest. Instead of finding evidence of viral DNA remnants in hosts, this paper makes a claim of physical evidence of old viruses, a, quote, fossilized virus, unquote. The paper entitled Fossil Evidence of Insect Pathogens from the Journal of Invertebrate Pathology is at, and gives a link. This, unfortunately, is a pay site, grumble, and wants $31.50 for the paper, double grumble. I want to read a whole slew of articles and can't justify paying $30 to $40 a piece on the chance they may contain something interesting, so I've been unable to read this paper. As far as I can tell from the abstract and the figures, which you can look at for free, the authors are making the case that based on their shapes, viruses were trapped with their host midge in amber. My question is, based on the figures, and if you can access the journal and the text, do you think their contention is valid? It's not real obvious to me that the objects in the pictures are viruses. Thanks for your help with this. Keep up the good work, David. So I had a look at this. And first of all, we'll probably get follow-up on this, but I question the use of the word. To me, fossil is something that has actually chemically changed in a way. Fossilized is it's become mineralized, okay? And I'm not sure that something trapped in amber fits that description, but I'm probably wrong. At any rate, these were insects trapped in amber that were investigated microscopically at basically a cellular level, and they found evidence for infection with a variety of different parasites, including fungi and etc. And their evidence for infection with viruses consisted of seeing what they interpreted to be inclusion bodies consistent with polyhedra. So they're not seeing, if this is true, and from my look at the figures, it's perfectly reasonable. The images that they were seeing are not inconsistent with this. They were seeing inclusion bodies in the cells that had a very regular structure consistent with being what any virologist would call a polyhedron. So this is for nuclear polyhedrosis viruses. These are insect viruses like baculoviruses, large DNA-containing viruses, where the virions, you make mature virions, and then clumps of these virions are included in yet another structure that is a single protein, that's the polyhedron protein, that's made in huge amounts and more or less crystallizes around these virions. So you get these super large chunks that have a crystalline-looking structure in the cells. And this is a way for preserving these viruses in the environment. When the host dies, these polyhedra are shed. They're very resistant to environmental insults. They're picked up by larvae that are feeding on contaminated material, and the polyhedra break down in the gut of the insect and release the embedded virions, which then infect the larvae. So that gives you an opportunity to see evidence for a virus, not by individual virus particles, but by the presence of these large inclusions. And my read of the they don't have any specific, protein chemistry or antibody labeling or anything like that. But based just on the microscopy, I think their conclusions are not invalid. The good double negative, right? Perfectly reasonable.
These say these amber specimens are 15 to 100 million years old,
right.
And this one with the polyhedra is 100 million years old.
Right.
Wow.
Right.
They looked good to me. Yeah.
Mm-hmm.
They look
good to me too. Yeah.
The authors on this paper are very interesting. We have George Poinar, who's at the Department of Zoology at Oregon State University, and then Roberta Poinar, who is at the Amber Institute, also in Corvallis. That's a cool place, the Amber Institute.
Yeah. Mm-hmm.
They should, they should get DNA out and see if they get viral DNA, right?
Confirm it that way.
Maybe they should make a dinosaur from these things.
There you go.
Yeah, it's pretty good. It's a nice find there, David. It looks good to us.
Yeah.
My next, yes,
Yep.
I guess so. It's from Patrick. Patrick writes, Dear TWiV team, I recently watched a nice TED Talk on science education / communication by Tyler DeWitt. The main thesis is that by making science fun and approachable, you're doing a service to the greater community, even if some of the details are lost. I'm often annoyed at the gross personification and oversimplification of science, particularly in popular science reporting. For example, imprecise metaphors often allow misleading, false interpretation, e.g. flu tells time. I know what that one's from. Mm-hmm. For these reasons, I found myself conflicted during his talk, but ultimately agreeing with the speaker's point that at the end of the day, it's more important to garner an interest in science and a sense for data-based reasoning than to drill down on the details to the point where the concepts are inaccessible. TWiV takes an alternative approach by inviting listeners to examine science and its intricacies through a scientist's perspective. I think there's room for both. DeWitt has contributed a number of short videos in an effort to make science more accessible. He provides a link to those on YouTube. His efforts are commendable and worthy, I think, of a TWiV pick. I also wonder if you'd consider doing an episode on science education and outreach. I think a discourse on how to solve the information gap is an important topic, particularly for a nation where science literacy is astonishingly low. Thanks, as always, for all your efforts. It's currently 40 degrees Fahrenheit with clear skies in the beautiful Pacific Northwest. Patrick, P.S. I recognize the irony in my comment about metaphors, since our lab, myself included, often use an arms race to describe host virus evolution. Patrick is in the Harmit Malik lab over at UW and the Hutchinson. It's okay if it's correct. When the science is presented incorrectly, then that's a problem, even if it's presented in a fun way. Mm-hmm. But you can make it fun and be correct, right?
Right.
So I didn't find this TWiV talk, the TED Talk. He didn't put a link to that, but I guess he didn't mean to. Anyone check that out?
I went to the YouTube, and I watched the one on valence electrons in the periodic table, since we had just talked about that with regard to your son memorizing it.
Right.
And I think it was 16 minutes long, and I was hooked for the whole thing.
Good.
good review of valence electrons.
So they have the screen is split, where half of the screen are his hands on a figure, and then the other half is him, right? So you can see his emotions and his interest in it. And he's very animated.
If you actually, I can get the link to the TED Talk. If you go to the TED site and
I'm sure we could find it there.
You can search him. I got it here.
Didn't we have another pick from someone who also makes these educational videos? Someone
seems very similar. If it's not this same person, it's somebody very similar.
It
but
was
it
a
might
high
even
school
be
teacher. Yeah,
that's right.
Yes, that's right.
Maybe in Bozeman, I think that was.
That sounds right.
Yeah.
I don't know which TWiV it was.
Good memory.
But
arms race. Yeah, that's problematic. But
but we understand what it means.
Yeah, I mean that's okay. But then there are people who talk about the front lines and combat and deployment of it's just too much. Just arms race alone is fine. Yeah. Thank you, Rich, for putting that in.
One of my close colleagues today, who shall remain nameless, was talking about some experiments today where they were interrogating the database.
Okay, who's next? I've forgotten. I don't know why I can't remember. That was me who just talked.
Then it's Kathy's turn.
Kathy.
Okay. Sijun writes, "Howdy, Vince, Rich, Alan, and Kathy. Hope you're all doing well. Sorry it has been a long time since I wrote in, but I promise that I have been faithfully going to the Church of TWiV whenever I have free time. But being in grad school and all, I'm not supposed to have any free time. I just came across an article published today and thought it would be very interesting to talk about on the show. And I don't recall what this one was.
The CRISPR and phages.
Yes.
The CRISPR and phages paper,
yes.
Right. We all know viruses have to hijack host cellular factors to proliferate while the host tries to fight back with antiviral factors, triggering an endless loop of coevolution between the host and the virus. This paper is interesting because it is the first to show that viruses might be able to not just steal elements of the host immune system and use it back against the host immunity. Just a side note, the CRISPR-Cas system that they looked at in the paper has recently been really hot as a potential new genome editing tool, in addition to ZFNs and talons. I can't remember what zinc
fingers,
nuclease.
Talons is another spin on the same thing.
Right. Okay, enough adrenaline rush. Now back to my boring life of reading papers, doing experiments, and writing code. Just kidding. Take care and keep twiving. Cheers. Sijun.
So I have not read this phage CRISPR paper yet,
it's
but
cool.
we need to do this.
Yeah, we did it in my class for Viruses in the News, the first 5 minutes of class. But I had to spend about 2/3 of that time telling them about the CRISPR-Cas system in bacteria. So, but basically, Rich, it's a system where there's the bacteria have found a way to put little sequences in that correspond to invading DNA from phages or plasmids.
Right.
But in this case, the phage has picked it up, and where those phage sequences would be, where there's foreign DNA sequences would be in the bacteria. Now in the phage, they're the bacterial sequences.
Oh.
So, yeah, it is really cool.
We did a CRISPR episode.
We did a CRISPR episode
ago.
a long time ago. Let's do this paper.
Yeah, we'll do it. I've been debating whether to do it here on TWiV, but if you guys,
I see. Okay.
but if you guys both would like to do it, we'll do it on TWiV. It's fine. It's not a problem. I'm going to do a TWiV at ASM in Denver this year, and Camille, the PI, is going to be there. I'm thinking of having him there too, so that would be cool.
You're going to do a TWiV at ASM?
Yeah, I'm going to do both.
Oh, good, because I'm going to be there. So
you are?
Yes.
I didn't see your name on the program.
Oh, I'm not on the program. I'm the DNA, whatever that subsection chair, that unit.
Right. Oh, so you come on to TWiV with me?
Sure.
It's going to be on Sunday at like 2 p.m.
Sure.
2:30. You be there then?
Sure.
Yeah, for sure.
I haven't made any plans yet.
So I already invited Nels Elde to join us.
Excellent.
Right? That would be cool.
Yeah.
He said yes. And I was just looking for one other person. This Camille is possible. Do you have the program, Kathy? You can take a look at it.
I don't have the program.
I'll send you I'll send a PDF and we can. Oh, that'll be fun. Cool.
Okay.
I was thinking of actually doing a real TWiV at ASM where we talk about papers instead of interviewing people. We'll see what happens. All right, cool. Rich, are you next? Yes.
Yes. Adam writes, "Hi, guys and gal. This is just too cool. Nothing more to say except that you couldn't make this stuff up." Is this the CRISPR paper?
Yeah, it's a CRISPR paper. Yeah.
Adam, 37 degrees C, 98.6 Fahrenheit, Melbourne, Australia, 24 degrees C, and sunny.
Oh, Adam is human temperature. Cool.
Yeah, good for him.
Glad you're not sick.
I get it. Okay. Yeah, that's right. You couldn't make this stuff up. Well said.
Yeah, that's good.
And Raihan writes, "Greetings to my favorite teaching team in the world. I came across this article from Twitter and would like to share it with you guys. It's a news report, and I'm unsure if it's made its way to the scientific literature yet, but if it's true, it sure is going to be a game changer." And this, of course, is the HIV cure in the baby that we just talked about. I'm still remaining skeptical, but this may be, it's just an overhyped story of how antivirals can control and suppress HIV infection. A fact I learned from you guys. That is to be skeptical. Keep up the amazing work. Raihan, the struggling PhD student from Singapore. So as soon as this paper comes out, we'll do it.
Yeah.
And maybe get Kathy to participate. Not you, Kathy, but
Kathy
the other.
Collins.
Then we could have two Kathys from Michigan.
Yes.
That'd be good and
That would be
confusing.
cool.
Yeah.
Kathy, you're next.
Patrick writes, "Dear TWiV team, I'm sure you've all seen this story, but if you haven't, I wanted to send it along. The New York Times article, again, the baby cured of HIV, and the first description of the data at the CROI. I think there are many reasons to be cautious regarding the results and implications of this single case, but there's no harm in being cautiously optimistic. I do worry a bit that this will be overly or inaccurately publicized. It also seems like a good opportunity to point out that even if these results lead to a real functional cure for HIV-1 positive infants, mechanisms and funding to get drugs to areas of the greatest need globally is sorely lacking, at least that I'm aware of. All the best, Patrick. P.S. Pretty gloomy right now in Seattle, 44 degrees Fahrenheit raining, but days are getting longer, and this story definitely potentially brightens things up a bit.
It's the same Patrick that we just had, right?
Right.
I should have grouped all these
That's all right.
thematically,
Okay.
but I did
And
not.
it's interesting because, of course, he sees 44 degrees Fahrenheit and raining as pretty gloomy, but that's warmer than it is here.
It is, but the rain is the hard
The
part.
rain is the gloomy part. I agree.
Vitamin D is probably pretty low in Seattle right now. Right? Rich.
Richard writes, "G'day, Twivernators." This must be from down under. "I am a paramedicine student in Melbourne, Australia. I was very excited to see that Vince was coming down south next year. If you are speaking anywhere that I can access, I'd love to attend. I have listened to your shows for a couple of years now, and it has hugely broadened my interest and knowledge in virology and microbiology in general. It is great to hear about non-clinical application as most of what I see as symptoms and signs, etc. Thanks for your weekly insight into these amazing tiny worlds. Where are you going to be, Vincent?
I think I'm going to, so it's ASM, but it's the Australian Society for Microbiology, which I think is in Melbourne. But I'm going to make a trip of it and visit a couple of other places. I think my family wants to come. So we'll get in touch with some other TWivernators down there, and we'll hook up. It's 2014. That's a ways off. Our next writer is Dixon Despommier, who sends a Yahoo News article. 2,800 pigs dumped in the Shanghai River raises concern. Now it's up to 13,000, actually. And this is an interesting story, which I just wrote about yesterday, actually, on my blog. And apparently they're upstream of Shanghai. There are lots of pig farms, and a number of them had an illness, and the farmers dumped the pigs in the river, and they floated down to Shanghai. And this is a problem because some of the water from the river goes for people to drink. And then they apparently sampled these pigs and the water, and they found circoviruses, porcine circovirus, which can cause illness in pigs, and which might have done so here, but we're not sure whether it did or not. This doesn't infect people. In fact, you may remember that the rotavirus vaccines are contaminated with these porcine circoviruses, and they're fine. So it's an interesting story.
It sounds to me like the big problem is farmers and others dumping sick pigs into the river.
Yeah, they shouldn't really do that.
Yeah.
So after I wrote my blog post, Alan Dove made a comment. He says he said that there's probably other viruses involved, and he closed his comment by saying, investigative journalism and transparency about public health aren't exactly China's strong suits, so it could take a while for all of the relevant facts to emerge.
Well, it's interesting because I hadn't heard about this in the news, but it came up at a dinner, and one of the people there was a Chinese woman, and she said that it's probably because the farmers are too poor to really dispose of them in any other way, so they just did whatever way they could.
Right.
I think that's
the
probably
most
right.
expedient way.
By the way, all the pork we consume has circovirus sequences in it.
Okay.
I read a paper yesterday from Ian Lipkin's group, which says most Americans have, if you eat pork, you have circovirus in your feces. You're not infected by it, but you eat it and it passes through. So all the pork has it in it. There are two circoviruses of pigs, 1 and 2. The 1 is not pathogenic. The 2 is. So even in a healthy herd in pigs, you can have one and it gets into the pork and it's no problem for
We eat all kinds of viruses every day.
Oh, yeah. Your cabbage is full
Cabbage is full of baculovirus.
Mm-hmm. Yeah.
Probably got polyhedra in it.
Yeah. It's cool. It's probably good for you. It probably stimulates your...
Sure.
Next one is for Kathy.
Yes.
Lynn.
Okay. Lynn Enquist writes, it came to my attention that you had a TWiV on DNA sensors in the nucleus. One of my students listens religiously. I think that you did not see a recent paper by one of my faculty, Ileana Christia, about her work identifying an interesting viral DNA sensor in the nucleus. I've attached it for you. I'm in China, Hangzhou, coming home today. You should do a TWiV on the 10,000 dead pigs in the river that flows through Shanghai. Looks like some of them had a porcine circovirus infection. Lynn. So, so I found this paper, and but that was late last night, and then I didn't go check it out today. But Vincent, it looks like you've had a chance to look at it.
Yeah, so she identified an interferon-inducible protein called IFI16, which is a nuclear sensor for DNA, for viral DNA.
Excellent.
So I think, and there is an email coming up later if we get to it, we should do these papers all together because this is really cool. This makes sense to me as a sensor.
Sure.
Right? Mm-hmm. Primarily in the nucleus. So it says we demonstrate that IFI16 detects herpes DNA primarily in the nucleus. It's really cool. And this kind of is, I wonder what the relationship between this and C-gas is, right?
Right.
If any. So we'll get a chance to look at that. I think that's cool. I like it. I had missed that, or maybe I had seen it and not paid attention.
I had missed it.
But here I was complaining about the leakage hypothesis, and now this makes sense, so we'll check that out. Rich, you're next.
Cedric writes, Dear Vincent et al., I'm not sure if this has been suggested before, but I would like to recommend a Chrome extension that stops YouTube videos from automatically playing, and gives a link. There's no longer a need to live in fear of a link suddenly playing audio while you're recording an episode.
This is brilliant.
Yes.
I never click a YouTube link because it starts playing.
Right.
Of course.
So we'll use that.
That's a great idea, Cedric. Thank you. Alan would like to know that, too. Next one is from Sandra, who writes, Dear Vincent, I don't remember where I first heard about TWiV, but I subscribe and have been listening to the podcast for a few months now. I have a BA in economics and an MBA and have worked in corporate America for over 20 years. In my late 20s, I discovered the work of Stephen Jay Gould and read everything I could find on natural history, including bios of Darwin, Wallace Huxley, and other great minds of the 19th century, and current work by great authors like Neil Shubin. I guess to summarize this, I have a strong interest in science, but absolutely no formal science education, just knowledge I've picked up by reading science in my spare time. I enjoy TWiV immensely, but find myself writing down terms used on the show with the thought that I need to look them up sometime and start to learn about cellular biology in order to get more out of the show. I could buy a textbook, but it has occurred to me that I might read an entire intro book on cellular biology and then not get much benefit, if most of the discussions your roundtable has are of concepts that would only be taught in graduate-level courses. With a full-time job exercising, keeping up with the economic, political, tech, and science news via the Internet, having a social life, and listening to TWiV and the Naked Scientists podcast, which recently had a podcast on epigenetics, I don't have the time to try to learn advanced cellular biology. I would like your advice as to whether reading an introductory cellular biology book might be adequate to allow me to get a lot more out of TWiV podcasts, or whether I should just stick to scribbling notes and looking things up. All right, so she then gives a pick, right? No. In return for the favor of your advice in this matter, I promise to never again tell anyone the flu shot is safe because it contains dead virus. Furthermore, I pledge to always correct anyone else who I hear making this mistake. Best regards, corporate American with zeal for learning science. P.S. I apologize if I have committed a faux pas by addressing this email to you using your first name. I have listened to so many episodes of TWiV now that I feel as if you, Alan, Rich, Kathy, and Dixon are a group that I am privileged to hang out with from time to time, eavesdropping on your thoughts about viruses, cellular biology, chemistry, etc. What a nice email.
That's very nice.
First name basis is fine.
Yeah, of course.
That's good.
It propagates the idea that we're just having a conversation.
Yeah.
So, yeah.
Do you guys have any recommendations to help her on cell biology? Basically, the biology of the cell is what she wants to know about, right?
Right. No.
No.
I mean, we need to write these books, Vincent. Okay?
We could do podcasts on them. I mean, we could do 5-minute, really simple videos on basic cell biology. How's that?
I mean, the books that come to mind are the standard graduate texts, right?
Right, but wow.
They're pretty heavy duty. And
Yeah.
if I were going to use those, I would buy one of those, and the names are escaping me right now. There's the Alberts.
Yeah, there's Alberts and, I got them right there. And Lodish is the other one.
Right.
Yeah, those are too hard for her. It's too big. They're huge tomes, and she'd get just turned off by them.
The only way I would recommend those is that if you had those, they would serve as they could serve as reference text. The problem is they may not work, okay, because they're just too dense.
Did Sampairak write a cell biology book? So
I don't think so. I know he's got the immunology one, which is good, and how cancer works.
Yeah.
I don't think he has a cell biology one.
Did Carl Zimmer write one? Let's see. Well,
But I think in the meantime, writing down the concepts and then looking them up, that's fine. I mean, that's the way we learn dictionary words or anything. I mean, if you don't go look them up later, then you're not going to learn it. So
Wikipedia.
Yeah.
Actually, a number of listeners have said that they do that as they're listening. They just Google it, and that really helps. Of course, that's what we do here.
Yeah, and the learning process is really interesting because in the beginning, it's hard to piece all this stuff together, but slowly but surely stuff starts to fit together, and you create a framework, and then the new stuff as it comes in is easier to fit into that framework, and it gets easier and easier as you assemble the framework. That's why it would be nice to have some sort of simple reference as a background that would provide some of that framework, but I don't know what it is. I got to write it.
You could try my students. I have a student, Ashley, who has a podcast. It's called Biochemistry Revealed. It was a pick of mine a long time ago. And unfortunately, they've stopped doing it. It was her and another student here, and they stopped at about 12 or so. And it's understandable. They didn't have the time to do it. But they actually did a few with some good basic stuff. If you want to check those out, they're pretty accessible. All right, let's see here. What should we do? We have a few left. I think we've been recording for an hour only, right? Yeah, we started 15 minutes late. Oh, it's 4 o'clock.
My watch says 5:17.
We've gone for an hour and a half.
All right, so let's wrap it up and do some picks, okay?
Okay.
Kathy, what do you have for us today?
Okay, so I changed my pick at the last minute. A friend of mine had sent me 2 articles from The Guardian that are more of the human backstory to the coronavirus EMC. And one of them is really just telling about the family and what happened. Is this is a recent case including 2 residents of the UK, and the father had gone to India and decided to stop in Mecca on his way back to pray for his son, who had learned of having brain cancer in November. When he got back to England, he was already coughing and probably gave the virus to his son, who has since passed away, and then the father is in a coma.
Wow.
So that's part of the story. But then they also talk about the physician who first identified the virus back in Saudi Arabia back last summer, or even before that, and so that's picked up in both of these stories by the same author in The Guardian, Ian Sample. And so the second story tells something that I was not aware of, that this first physician reported his findings on ProMed, and then in very short order was fired from Saudi Arabia and is now back in his home country of Egypt because they didn't like him spreading the news about this disease in Saudi Arabia.
Wow, that's too bad.
So, and then the second article goes on and talks a lot more. It interviews, I think Fouché was in there, talking about how there needs to be a lot more transparency about this, and people need to be doing the epidemiology and that part of the world, and that's maybe not happening in the way that it should. So I just thought it put more of the human spin on the science that we'd been talking about the last couple of weeks.
Mm-hmm.
You know, there are some governments in this planet where transparency will never occur, even with something that's so globally important like this, right? It's unfortunate, but it's happened before, and it's going to happen again, unfortunately, because, this information needs to be free, but, this stuff like this happens.
Right.
It's bad. Rich, what do you have for us?
I have a book that I just read. It is called The Self-Illusion, How the Social Brain Creates Identity by Bruce Hood. This is sort of a kind of a popular psychology book that addresses a theme that has been of interest to me for a long period of time, and that is, what is this thing that we call self? What is me? What is I? What is myself? And the bottom line here is that self is a concept. It's something that we basically make up as we develop in order to basically interface with the rest of society. In fact, the idea that self is just a concept, and the illusion is a strong word, okay? But concept is easier to swallow, especially for the Western mind that is so fixated on self. This is actually an idea that goes back thousands of years. There are Eastern philosophers from 2,500 years ago who recognized from their own investigations that self was, the strong way to put it is that it doesn't exist, all right? But at least that it's a concept or an illusion. And this is sort of a modern psychologist's spin on the same topic. So it's got a lot of psychology experiments in it and stuff that talks about child development and how personality develops and et cetera, and how the whole concept of self develops in somebody and what its function is. And I found it sort of an interesting read.
Cool. I think we're just parasites, man. Parasites of Earth.
Yeah. Oh, yeah.
Or we're mostly bacteria.
We are mostly bacteria, yeah. Wow, that's cool. Should I buy it?
Sure.
I would like it?
Yeah, I think you'd like it.
Okay. Well, that's why you're recommending it,
if you have not considered this idea before, it could shake you up a little bit.
I don't, the stuff I read is never about that sort of idea, what is self and what makes you and all that. It's too heavy for me. I like viruses. It's factual. Well, of course. My pick is a recently released report by the American Academy of Microbiology, of which you now are a member, Professor Condit.
And of which Dr. Spindler is also a member.
Is that right?
Of some longer standing.
You got in a while ago?
Not much longer.
Oh, I'm sorry. I didn't know that. Congratulations to you, too.
Thank you. That's a while back.
I was on the first meeting this past year. I don't know why I'm on this. Oh, I was asked to be on the elections committee. That's right. So I know everyone who's been elected. And so they do these colloquia. One of the things the Academy does, besides collecting famous people like Spindler and Condit, is they publish colloquia. They have colloquia. Colloquia are meetings where they get people together to talk about a topic, and then they make a report out of it. And this one is cool. It's called Moving Targets, Fighting the Evolution of Resistance in Infections, Pests, and Cancer. So it's all about antibiotic and resistance in different aspects of our world. So it's cool. It's a free report. You can download it and just read it at your leisure. So check it out.
Excellent.
Very cool. We had a pick from Liam, eLife, and Rubric. And then we have one from Steve, who writes, Hi, TWiV team. Having listened to each and every TWiV podcast since the inception of the show, you have tantalized us and yourselves with the idea of inviting a guest to the show to talk about plant viruses. I am a retired emergency medicine physician, so have lots of time to listen to science podcasts. In addition to TWiV, there are other excellent science and medicine podcasts that I listen to, including AAAS's weekly Science Magazine podcast. At the recent AAAS meeting in Boston, they interviewed Dr. Marilyn Rusink from Penn State University about plant viruses, and that's my suggested listener audio podcast pick of the week. And he gives a link to that podcast and tells us how to find it. Having listened, don't you think Dr. Rusink might be an excellent TWiV guest? She is knowledgeable and clearly loves and knows her field. Equally important, she has an enthusiasm for a subject that is infectious, and she gives a great interview. She is poised, articulate, and relaxed. Puts a link to her webpage, and then he says you don't have to read this on the show. And I'm happy.
That never works.
I'm happy just knowing that I made a contribution to your show. No need to read my email. Warm regards and thanks for the many hours of superb podcasting. Science is just wonderful stuff, and listening to scientists who enjoy doing what they do, like the TWiV team does, is just so special. I am so glad I spent my career in the sciences.
Isn't that nice?
So we had Marilyn on TWiV number 92, Steve. Check it out. Live at ASV in Bozeman. You remember that?
Yep.
That was our first ASV episode. And I thought she was very good back then.
She was very good. We ought to get her back
I'd like to get her back now that she's at Penn State. It's not too far away.
Yep, get her back again. Good idea.
We're going to be at Penn State this summer.
Yeah, I keep getting emails from them saying that they've made an account for me. Somebody must have signed me up for this meeting.
Oh, I don't know why they do that. You do it yourself. I just signed up myself today. Anyway, you going to be there, Kathy?
Yeah, I haven't signed up yet. I'm just writing myself a note. You better do that.
Sign up. I just told my students because we're going to all drive there. Mm-hmm. I'm bringing my whole lab, and I figured I'd drive to save money. So we're gonna have a road trip.
Excellent. They have 2 great golf courses.
I don't play golf.
Well,
I'm sure you and McFadden will again.
yep.
You don't play either, Kathy, right?
No, I don't.
Good. At least I got company here on TWiV. I don't think Dixon plays. TWiV is at iTunes and TWiV.tv. And please go over there to iTunes and rate the show, leave a couple of stars, and that helps us to stay visible. And do send us your questions and comments to TWiV at TWiV.tv. Rich Condit is usually at the University of Florida, Gainesville, but he'll be back there tomorrow. Thanks for coming uptown, Rich.
This is just great fun. Good to be back in the TWiV studio. Having a great time.
You missed a few sessions, right? Yeah.
I did.
Welcome back.
Thank you.
I'm glad you came uptown. I know it's kind of a pain.
Oh, no, how often do I get here? This is fun.
Once a year, it seems now, right?
Every other year. With
that's right. The course is every other year. Are you going to be with us next week?
Yes, I believe so. I'm around for at least another 6 weeks, and then I take off for another little while.
That's right. Everybody is back for March 29th. Cool. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun.
And you'll be with us next week as well.
Yes.
I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.